Hello everyone, welcome back to my channel IGNOU DNHE online classes. I am Dr. Aayushi Choudhary. Let's continue our studies, and now today we'll be studying a very important chapter of DNHE 2, which is Unit 4, Agents of Contamination. This unit is central to understanding how infections originate, how they spread, and how we can protect ourselves and our communities. By carefully examining the agents, reservoirs, transmission modes, and host defense, we will see how disease is not just a biological event but a chain of interactions that can be interrupted with knowledge and preventive action. We will study today Introduction, Agents of Contamination, Sources and Reservoirs of Infection, Modes of Transmission of Infection, Mode of Entry into a susceptible host, Prevention and Control of Infection and Disease. Introduction. An infection occurs when microorganisms succeed in entering a host and begin to reproduce. A disease is the outward clinical manifestation of this infection. The fever, cough, rash, or other symptoms we recognize, all of us have experienced infections at some point. Sometimes they result from our own negligence, such as poor hygiene or unsafe practices. At other times, they occur without any fault of ours, as in the case of airborne diseases or congenital infections. While some infections are unavoidable, most can be prevented through awareness, hygiene, and timely medical intervention. Objectives. In this unit, we aim to identify the different microorganisms that cause infection, understand how infections are spread from one host to another, learn strategies to control and prevent infections, recognize the role of the environment in disease transmission, adopt practices that safeguard both individual and community health. Agents of contamination. Microorganisms that contaminate living bodies or inanimate substances are called agents of contamination. These include viruses, rickettsia, bacteria, fungi, protozoa, metazoal parasites, and ectoparasites. They are parasitic in nature, living at the expense of the host they invade. Once inside, they replicate within tissues and cause infection. For example, the influenza virus enters the human body, multiplies, and manifests as fever, headache, running nose, and sneezing. This illustrates the chain of infection, which requires three elements. First, the agent of infection. Second, the mode of transmission. Third, the host. The agent is the direct cause of disease or infection. The next link in the chain of infection is transmission, the mechanism by which the agent moves to a new host. The last link in the chain is the host, the organism into which the microorganism enters, settles, multiplies, and produces illness. When an infectious agent is present on a body surface or on an inanimate object or substance, this is called contamination. However, finding an infectious agent on a person does not automatically mean the person has the disease. It simply indicates a potential for disease to develop. Agents of infection or pathogens. Let's see these agents in detail. Viruses are the smallest pathogens. They reproduce only inside living cells using the host resources. They destroy host cells and evade antibiotics. For example, they include influenza, polio, measles, AIDS, and rabies. Rickettsia resemble viruses but are susceptible to antibiotics. They spread through bites of infected ticks, lice, and fleas, causing diseases like typhus and trench fever. Bacteria can multiply outside living cells and are visible under a microscope. They cause diseases such as cholera, tuberculosis, diphtheria, typhoid, and tetanus. Fungi. Fungi may live independently on a parasite, causing skin and oral infections. Protozoa are single-celled organisms responsible for malaria, amoebic dysentery, and kala-azar. Metazoal parasites are multicellular organisms like worms, hookworm, and roundworm are common examples. Ectoparasites live on the skin or external body parts such as lice and scabies mites. Sources and reservoir of infection. A source of infection is an entity from which a pathogen is transmitted to a new host. This can be a person, an animal, an object, or a substance. A reservoir is the environment in which a pathogen lives, multiplies, and depends on for survival. Reservoirs may include people, animals, plants, soil, inanimate substances, or combinations of these. While a reservoir is the pathogen's natural habitat, it is not only the immediate source of infection. The source usually refers to the immediate origin of the infectious agent and may be part of the reservoir or separate from it. For example, in hookworm infection, the reservoir is humans, but the actual source of infection is soil contaminated with infective larvae. For tetanus, both the reservoir and the source are soil. In typhoid fever, carriers, means the infected people, serve as the reservoir, while the source of infection may be patient's feces or urine, or contaminated food, water, or milk. There are three types of reservoir. First, human, second, animal, and third is non-living. Human reservoirs of infection. Humans are the most common source of infection for other humans. Diseases like an influenza, measles, chickenpox, mumps, polio, hepatitis, AIDS, and worms often spread this way. Human reservoirs fall into two main categories. First, persons who have the disease. In this, there are three types. First, clinical cases. Individuals show clear signs and symptoms. They are obvious sources of infection and can be identified easily. Second, subclinical cases. Individuals carry the pathogen but show no symptoms. These are more dangerous because they unknowingly spread infection while living normal lives. Subclinical infections are common in diseases like mumps, polio, hepatitis A and B, influenza, and diphtheria. Over time, repeated subclinical infections can help build immunity. And third one is latent infections. The pathogen remains dormant in the body even after apparent recovery. It may reactivate under certain conditions. Example, herpes simplex. And second type is persons who have the infectious agent in their system but do not show any evident disease or carriers. Some individuals harbor infectious agents without even developing disease themselves. They remain immune but can transmit infection to others. Carriers are less infectious than clinical cases but more dangerous because they are hidden sources of spread. Animal reservoir. Birds and animals are also sources of infection. The infections and diseases which can be transferred to men from birds and animals are called zoonoses. There are more than 150 zoonotic diseases that man can catch from birds and animals. Some examples are plague, rabies, yellow fever, Q fever, and kala-azar. Wild birds in particular play an important role in spreading the mosquito-borne encephalitis and several other mosquito-borne diseases. On migrating from one locality to another, they may carry ticks infected with viruses and rickettsia that may cause diseases in humans. Non-living reservoir. Soil and other inanimate matter can also act as reservoirs of infection. Soil may harbor agents that cause tetanus, anthrax, and mycetoma, and other agents can get into humans or other animals. Once the agents of infection can find a place to live and multiply in, they are ready to find a new host for passing on the infection or disease. This step is referred to as transmitting the disease. Diseases may be transmitted from the reservoir or source of infection to a new susceptible host in many different ways. It is important for us to know these ways so that by taking relevant precautions, we can prevent the spreading of a disease. This brings us to a discussion of the ways in which an infection gets transmitted. Modes of transmission. Infectious diseases spread through specific routes depending on the nature of the disease, though some diseases utilize multiple routes for transmission. For instance, AIDS can spread via sexual contact, blood transfusion, or from mother to fetus, thereby increasing its chances of survival and propagation. Below are the various modes of transmission. Infections spread through direct or indirect transmission. Direct transmission involves immediate transfer from source to host, such as contact, droplets, soil inoculation, or transplacental transmission. And second is indirect transmission. It involves intermediaries like contaminated food, water, vectors, air, fomites, or unclean hands. Direct transmission. Direct transmission occurs in several ways. Direct contact through touching, kissing, or sexual contact spreads diseases like AIDS and leprosy. Droplet infection. It occurs when saliva or nasal secretions are expelled through sneezing or coughing, spreading influenza or tuberculosis. Soil contact allows pathogens like tetanus and hookworm to infect tissues. Inoculation through bites or contaminated needles spreads rabies and hepatitis B. Transplacental transmission allows pathogens like rubella or syphilis to pass from mother to fetus. Indirect transmission. Here, the agent first exits the source and uses an intermediary to reach the host. Vehicle borne. Spread occurs via contaminated food, water, or biological products. For example, typhoid, hepatitis A, and malaria, in which typhoid is spread via water, hepatitis A is foodborne, and malaria is through blood transfusion. Vector-borne. It involves living carriers such as insects or animals. Mechanical transmission. Pathogens are carried externally, for example, flies on the food. Biological transmission. Pathogens develop within vectors before infecting host, for example, malaria via mosquitoes. Airborne. Its spread occurs via inhalation of droplet nuclei or dust, for example, tuberculosis, measles, and Q fever. Fomite-borne. Inanimate objects like clothes, utensils, or surgical instruments harbors pathogens, for example, diphtheria, dysentery, and hepatitis A. Unclean hands and fingers. Infections are spread by touching food, water, or wounds with contaminated hands, for example, typhoid, dysentery, and intestinal parasites. Next is mode of entry into host. Pathogens enter the body through different routes like respiratory tract, gastrointestinal tract, genitourinary tract, skin, and mucous membranes. And various diseases are transmitted through these. For example, respiratory tract, tuberculosis and influenza, gastrointestinal tract, typhoid and dysentery, genitourinary tract, AIDS and syphilis, skin, leprosy and rabies, and through mucous membranes is diphtheria and whooping cough. Next is prevention and control of infectious disease. The ultimate objective of prevention and control of a specific disease is to make it disappear from a particular geographical area. This is called eradication. For a disease to be completely eradicated, it must disappear from the face of the earth. The only disease so far which has been globally eradicated is smallpox. With the whole world being involved, you can imagine the complexity of the problem. Our efforts need to be directed at the agent at its source or reservoir with the objective of either destroying it at its very point or containing it till it dies itself due to lack of new host. Not allowing the infection or the disease to spread to a new host that is, interfering with all means of its transmission. Third, protecting the host by making him or her strong enough to resist the infection or disease. Controlling the agent's source or reservoir. The best way to control infection is to eliminate the disease agent at its source. It is easier with animals or inanimate reservoirs, but more complex with human reservoirs. Two key steps are early diagnosis and treatment, and second is notification of authorities. So, early diagnosis and treatment includes detecting and managing cases promptly, which prevents further spread, and notification of authorities, in which certain diseases must be reported by law. Isolation. Separating infected individuals from others remains one of the oldest and the most effective methods. Isolation may occur in hospitals, special wards, or at home with precautions. Quarantine. Quarantine prevents the introduction of infection into a healthy population. It applies to individuals, animals, plants, or even vehicles and goods. The quarantine period equals the longest incubation period of the disease. It is widely used at borders and points of entry to stop the spread of communicable diseases in the humans, animals, and plants. Techniques to destroy the agent. Destroying by heat. Boiling water, which kills bacteria in 5 minutes and most of the spores in 20 minutes. Next is pasteurization. It is used for milk products. Example, by heating to specific temperatures. Steaming. It sterilizes at high pressures, for example, autoclaves in hospitals. Dry heat. It is suitable for glassware and metal. Second is destroying by drying. Some bacteria die upon drying, for example, meningitis causing microbes, but spores may survive. Destroy by sunlight. Effective for minor infections using UV rays. Destroy by chemical disinfectants and antiseptics. Disinfectants kill organisms, for example, chlorine for water sterilization. Antiseptics inhibit growth without destroying organisms, example, iodine for skin. Common chemicals include phenol, alcohol, soaps for general cleansing. Last is removing the agent by infection by filtration. Filters remove bacteria from liquids but may not always remove viruses. Regular cleaning is required to avoid contamination. Next is preventing the spread of infection. Our effort to prevent the spread of the disease means that we would need to adopt techniques which would interfere with the transmission of the disease agent from the source to the susceptible host. These techniques will depend upon the channels and modes used by the disease agent for transmitting itself. In diseases which spread by direct contact, we need to direct our efforts to proper washing of hands and feet and isolating patients from others. Indirect contact transmission can be interrupted by sterilizing or decontaminating the objects associated with the spread of the disease. Separating items used by the sick from those used by others is another important step, for example, utensils, linen, or syringes, etc. In diseases that spread by droplet infection, wearing a mask while attending on the will help. Airborne transmission may be prevented by controlling the source or by using air filters in the air stream in hospitals. Adequate dust control and cleansing and dusting will prevent dust-borne infections. Vector-borne infections can be controlled by eliminating the vectors such as ticks, fleas, or mosquitoes. Next is protecting the host from infection and disease. Improving the host defense mechanisms can help reduce susceptibility to infections. Measures include natural defense mechanisms. Skin and mucous membranes act as physical barriers. Tear production, mucus secretions, and inflammation help against protect microorganisms. Genetic traits and general nutrition enhance immunity. Immunity types: natural immunity and artificial immunity. Natural immunity develops after exposure to an infection, sometimes for life. Artificial immunity induced through immunization. It is active or passive. Immunization. It is a key tool in disease prevention. Immunization reduces the population's susceptibility to infections, contributing to herd immunity. Active immunization introduces antigens to stimulate antibody production, providing long-term protection. For example, it includes vaccines for polio, measles, and tetanus. Attenuated vaccines, which are used by weakened organisms. For example, yellow fever. Inactivated vaccines use killed organisms, for example, in typhoid. Passive immunization. It introduces preformed antibodies for short-term protection, for example, immunoglobulin for hepatitis. Active immunization. Active immunization is one of the most effective and economical tools in modern medicine, forming the backbone of infection control for diseases such as polio, tetanus, diphtheria, and measles. It works by introducing antigens, either weakened live organisms or killed inactive forms, into the body to stimulate antibody production. These antibodies then protect the host by neutralizing pathogens or their toxins. Routine vaccination during infancy and childhood with scheduled booster doses ensures long-term protection and contributes to the herd effect, where widespread immunization reduces overall susceptibility in the community, sharply lowering disease incidences even if not even individual is vaccinated. Although immunization is not always 100% effective, especially when individuals are exposed to large doses of pathogens, it remains a critical preventive measure. Passive immunization. It provides immediate but short-term protection by introducing ready-made immune substances such as antibodies directly into the body. For unlike active immunization, this method does not stimulate the body to produce its own lasting immunity, making it less useful for mass disease control. Protection can also be enhanced through chemoprophylaxis, where antibiotics are given before infection occurs to eliminate pathogens upon entry. Thus, passive immunization and prophylactic drug can serve as supportive long-term strategies to reduce risk and manage exposure, complementing the broader long-term benefits of active immunization. Passive artificial immunity in newborns develops through maternal antibodies transmitted via the placenta, offering short-term protection for about 3 to 6 months. Beyond this natural safeguard, prevention of infection lies heavily on personal knowledge, awareness, and everyday hygiene practices. Thus, both maternal immunity and consistent hygienic behavior form essential defenses against a disease, reinforcing the idea that prevention begins at the individual and household level. Role of environment in protection from infection and disease. Environmental factors have a very definite bearing on each link in the chain of an infection, on source or reservoir, on transmission, as well as on the susceptibility of the host to catch infection. Temperature and humidity influence the growth and survival of microorganisms in the inanimate environment. They also affect the integrity of the skin and mucous membranes of the host. In dry cold weather, the skin and the soles of the feet, as well as the mucous membranes of the lips, get cracked, thereby allowing access to infection. Population density influences the ease with which infection spreads. The denser the population, the more easy for infection to spread. Pollution like cigarette smoke, which irritates the respiratory tract, may reduce the host resistance to diseases of the respiratory tract. Here are some important questions for exam point of view from this unit. You can go through these and prepare for exam, and also don't forget to solve the check your progress exercises given in the study material. In the next video, we will continue with Unit 5, Water Supply and Waste Disposal.